Hey there. I'm just going to start our exam 2 review. I'm going to go through kind of housekeeping items and then module 4 items. So before we start, let's see, here's a little spiritual thought for you. I served my mission in the Czech Prague mission, and I was changed by our president, who continually taught the atonement. He taught about Christ's grace. And I loved how he would say we have access to his grace before, during, and after all we can do. So as we're studying, we can plead for his grace as well, to help us to remember, Anyway, so I thought it went along with this little quote from the New Era. Whenever you feel alone or overwhelmed or discouraged, remember that the Lord Jesus Christ makes his grace available to you. I know it can be tough to learn all this material. It can be tough to balance school with work and family and callings and other things we have going on. But we always have access to his grace. All right, so with the exam 2, it opens on Monday, week from yesterday, and closes on Tuesday, February 12th. Closes at 11 p.m. Do not start at 10 p.m. Start earlier. Give yourself plenty of time to go through and be able to think through all the questions without having to stress about a 11 o'clock deadline. Similar format, or basically the same format as last time, 50 multiple choice questions. Be sure to study a variety of material and all the course material. As you go back through, there will be little details that stick out, and they like to sometimes grab some of those little details and stick those in as test questions. I love this little Peanuts comic. The more I study, the more I realize how little I know. And I think you can take that and use it to your advantage as you go through and study, okay, this is what I don't know. This is what I have questions on, and ask those questions on the collaboration board or take time to really dig more into those topics. And along with that, we have the extra credit. I'm going to skip to this lower bullet here. The extra credit, hopefully you saw that in the announcements. It really can be helpful to draw out concepts and to use those concepts, those drawings, to teach other people. One concept that I would recommend that you draw out, if there's not another one, if there's another one you're struggling with, this one, let's see, the action potential graph in 5.3 would be a great one to draw out and talk and teach somebody. So as you study, realize where your weak points are, and then use that to really make those weak points strengths, and hopefully this extra credit can help. I look forward to seeing your posts on that. With Proctorio, please don't stress about Proctorio. I had a visit with Peyton this morning. Hopefully you don't mind if I call out Peyton. I loved her attitude on Proctorio. I know a lot of times people get nervous because we've got a camera watching you. She said she loves taking exams where she's sitting in her own space, her own room or her house or wherever, and instead of in the testing center, and it's a place where she can be comfortable. So thank you for that awesome attitude on that one, Peyton. With Proctorio, again, remember to show the scratch paper before and after. If you forget, or if something happens, like somebody walks in, don't stress about those things. Basically what happens with Proctorio, they've got people monitoring a bunch of students on different screens, and they'll just email us if there's an issue that comes up. And so if I've heard from you already and I get a notification, I'll just disregard it. So no biggie. Don't stress about that. Again, make sure you understand the concepts very well. Spend lots and lots and lots of time with Module 5. So right now I'm just going to go over Module 4. It is represented on the exam, but if you notice, Module 4 is pretty small. Module 5 is big, and there is a lot of information in there. So don't wait, don't procrastinate getting into it and really starting to hash out those concepts. Because the bulk of the exam is concepts, applying concepts from Module 5. That said, there are several questions on organelles and cells. So we're just going to go through this really quickly. I know you can review this on your own. I know you've got flashcards. Hopefully this would be a really good topic to learn with flashcards. Or I've seen some of you write in your notes on one side and then you can fold it over and use that as a sort of flashcard. That would be really cool. Great too. So the 4 characteristic functions of the cell: cell metabolism and energy use, synthesis of molecules, communication, reproduction, and inheritance. So our precious DNA is housed within the cell, within the nucleus, and this has all the information we need for reproducing and making new cells. There's just an image of the cell. All right, getting into this, we've got the lipid bilayer up here. I know some of you missed this question on exam 1. Hopefully at this point you're very familiar with the lipid bilayer and that it is composed of phospholipids. And as you get more into 5, you'll learn more about the proteins or the channels that are here, and that will be really helpful. All right, the nucleus. The nucleus is where the DNA is. The nucleus protects the DNA, keeps it from getting damaged, because damaged DNA means altered structure, which means altered function for our proteins, which could cause all sorts of problems in the body. So again, that's the nucleus. Cytoplasmic organelles. So these are the organelles within the cytoplasm, or the rest of the cell. We've got the ribosomes, 2 little pieces of messenger RNA and protein that make up the ribosomes. These are the sites of protein synthesis. This is where proteins are synthesized or made. The rough endoplasmic reticulum is rough because it has all these attached ribosomes, and this is going to function to synthesize proteins and transport them to the Golgi apparatus. The smooth ER, smooth because it has no ribosomes, is going to manufacture or synthesize our lipids and carbohydrates, detoxify harmful chemicals, and store calcium. Note this stores calcium. So these are the kind of little details that they can grab. All right, let me move this. So the Golgi apparatus is part of the packaging and shipping. Little vesicles can break off and take the structures that they've made or that have been made and transport them throughout the cell or to the outside of the cell. Lysosomes have digestive enzymes. And if you look back into the text and if you look on the review guide, these digestive enzymes can break down macromolecules. Peroxisomes, similar function. Proteasomes, all these guys are in that breakdown family. Mitochondria, we're going to talk about them more in a few minutes, but basically their big function is to synthesize ATP or energy. They're the powerhouse or the energy suppliers of the cell. The centrioles are, notice right here where it says cylindrical organelles in the centrosome, and this is part of the cell that is involved in cell division. Let's see. Cilia and microvilli we really didn't talk a whole lot about. All right, mitochondria. They have an outer and an inner membrane that we're going to get more in detail with when we get into metabolism. But this inner membrane has all these folds or cristae, which gives a greater surface area for protein, or sorry, not protein, ATP synthesis. I get thinking synthesis, just program, program protein. But ATP synthesis, we're making ATP in the mitochondria, or the powerhouse of the cell. The endoplasmic reticulum, we have the rough endoplasmic reticulum that we talked about already with the ribosomes attached. And this is where protein synthesis is happening. These ribosomes synthesize proteins, they go right on in. And from there, they are packaged and shipped in the Golgi apparatus. And then if we take a look at our smooth ER, packaging and synthesizing proteins, lipids, steroids, and storing calcium. All right. I think this is about where we'll wrap up with this one. The big central dogma of biology. is that we have DNA, we have this genetic code housed within the nucleus that is used as a template. As the DNA will kind of pull apart, we'll be able to use this template to make messenger RNA or to transcribe into messenger RNA. And if you think of transcription, so like with if somebody's transcribing a speech, they're writing it down. It's in the same language. It's still in the language of nucleotides. And messenger RNA is still nucleotides. But when we go to protein, we're translating it into proteins, into the language of amino acids. So this is the translation. And this is the central dogma. So the protein then is moved into the ER, then packaged and modified, shipped by the Golgi apparatus, and then taken out to wherever it's needed. So anyway, happy studying. Watch for more videos to come.